welcome to the lecture 16 of the learning about learning lecture series. And till now what we have seen or more specifically in the last lecture what we have seen is that how molecules as small as dopamine, a very small molecule, can actually control the levels of dopamine, can modulate and control the expression of a behavior and then switch an animal from a sign tracking versus to a goal tracking and so on and so forth. Is just to illustrate a point of how a complex behavior can be controlled by individual molecules. In line with that, what we are going to see in the coming lectures are that we are going to take this idea of complex behavior tracking down at multiple scales, and while we are doing that we will see that it is not just the molecules, the behavior can be at multiple levels. It can be at the level of neuronal networks, it can be at the level of synapses, the junctions through which the neurons communicate, it can be at the levels of the molecules as we have seen already. In doing so, for us to do so requires few preliminary few preliminary information and a knowledge, and we would gather them as we go along. Now remember our very first lecture. In the very first lecture when we talked about memory, when we talked about memory we said memory has different phases and these phases can be separated by using selective drugs, right, by infusing drugs at several different points in time, and one of them was protein synthesis inhibitors, anisomycin. Now let us take I promised you at that point in time we will look at it in a little bit more deeper manner later in the course, and that is the time now. Just to rehash, at that time what we said was that if you train the mice, a group of mice in a task and then infuse protein synthesis inhibitors, in the specific case of that study we said anisomycin, and then infuse this drug, test them like half an hour or 1 hour later, you would see that their memory is pretty good compared to the control. They are indistinguishable. However, if you were to do that test 24 hours later, 24 hours after the training, and right after the training you are infusing the anisomycin of the training, but either way we were talking about infusing them right after the training, and we said we will not see this memory expressed at all. How did that result come by? Originally that result or that experiment was done by Agronoff et al. Shown in the slide here, and they used goldfish to study this process, to study the effect of protein synthesis or the usefulness of the protein synthesis in memory consolidation. So what did they do? They took a behavioral paradigm wherein they will take the goldfish, they will use the goldfish, and then the fish obviously swim in water. So there are two chambers in this fish tank, and one chamber is well lit, nice, and other chamber is dark. There is a connecting path, all right, there is a connecting path between these two chambers, and as you can see the way I have indicated the water level in blue, the water levels in the tanks are pretty good, the fish can actually swim all around. However, right at this point where there is a bridge that is connecting these two chambers, the water level is just about bare minimum. Basically this possesses a hurdle, for a barrier so to speak, for the goldfish to go from one place to the other. You can actually put some physical barriers too, but the idea is essentially the same. You want to make this crossing the bridge a slightly difficult task. It is a possible task, but it is a difficult task. once you have done that, then what they are doing, what they are going to do is that they are going to do the CS US pairing here. So, what is the CS? The CS is a light coupled with electric shock. Okay. Right before the electric shock is given, the light goes off, that is the CS. The CS comes on, then it goes off, and then the electric shock is given. Nobody likes to receive the electric shock, so does the fish. Then in response to that, it needs to take an evasive action. It has to escape from this chamber all the way into the bridge to the dark box. In this place, if they go in, there is no electric shock. Clearly the animal does not learn it at the very first instance. They had to do this in a block of like 10 or 20 trials, and every time when they are scoring, when they are giving the training, they are going to score whether the fish successfully went from the shock box to the or the light box to the dark box. If it did, then you would say that okay, it successfully exhibited the learning. If it did not, then you say it does not exhibit the learning. But that is about learning. Here we are talking about memory retention or consolidation to speak. Why are we what is the distinction here? The distinction is we have to test if the animal remembers something, and when we are testing if the animal is remembering something, typically we do not give the US, we just test the animal's response to the CS alone. So, on the test day you are not going to give this electric shock, but you are going to expose the animal to the CSQ, this is the CS, remember, and we are going to test the animal to the CSQ and then ask, can the animal remember the association that it developed during the learning between the light and the shock. If it did, it is going to go into the dark box. Again you do it since it is a 0 or 1 response, you do it in multiple trials and then get an average score of how many times actually the goldfish went from the light to the dark box. Now you compare this with the trials that are done during the training, they get a score something called as a retention score, that basically is the difference. If the difference is low, then what you say is that the animal is not retaining anything at all, as it is high negative and low in total value. And if on the other hand, if the animal really retains well, then you would see that they remember this. So, they would elicit a response pretty nicely and go to the dark box, not very different from what they have initially started with. So, we remember this is equivalent of our comparison of the 30 minutes or short term memory with the long term memory in the mice experiment. Here they are comparing after the training trials, I mean in the later training trials with that of the retrieval trials that are presented 4 days later. Okay. What they but in between what they are going to do is that they are going to divide the animals in to groups. Control, those guys receive just the vehicle or the saline, and then the other guys receive puromycin, another drug, another protein synthesis inhibitor, and that protein synthesis inhibitor is injected into this animal. The reason why they were actually doing this in the goldfish is because it is easier to access the cranial cavity in the goldfish. Actually they all that they have to do is they have to take that a needle, just a syringe and a needle, just like what we do, what we what we use for giving a shot to ourselves, or a doctor uses to give a shot, and then go and then directly inject at the cranium, all right, at this in this place. And when you do that, fish really recover very fast. I mean in few minutes they do not exhibit any signs of having been injected, and then you can subject them to the test. So, they did that right after the training is over, and then waited for 4 days and asked how well are they doing it. They did even slightly better. What they did was they measured the dose response curve, and they clearly show as a function of different dose the ability to retain or ability to lose changes, and in this particular case 2 of such doses has been shown, one with the dotted line, other with the solid line. What you are actually looking at is the graph of when they injected as a function of time in this axis. This is the time elapsed since the training. Okay. It is not coming out nice, so let us write it here. So, it is the time elapsed since the training, and then you are measuring the animal's ability to recollect on the 4th day. Now, what you can see from here is that if the injection were to be given right then and there versus 360 minutes later, right then and there you inject, clearly they are performing less compared to a test that was done right after the training. Okay. That is what the retention score is measuring. If it is more negative, it means on the 4th day they are performing less, that is why it is negative. So, it is a 4th day performance minus the 1st day performance. So, the retention score you can think of that as 4th day minus the 1st day, alright. So, you can see that more negative here means that the 1st day performance was better, the performance has declined, their ability to retain has come down, that is why it is negative, and that is that even that is dose dependent. The amount of puromycin that you inject actually pushes down the performance levels. As you increase the time until about 90 minutes or so, there is some effect, but anything after 90 minutes, particularly at 360 minutes, it is about 6 hours or so, you do not see any difference, it is 0, it is the performance is as good as the 1st day, right. So, the consolidation has taken place, or at least seem to have taken place at 6 hours. Any time afterwards, if you try and infuse this protein synthesis inhibitors, it is not of much use. Since then, lot other more experiments people have done, and what we know is that if you were to be safely out of the consolidation domain, then it is good to wait for about overnight or 24 hours to be safe. Anything greater than 24 hours you can safely assume we are out of the protein synthesis dependent consolidation phase, right. So, it is all that is all happened, and if you want to intervene before that, it is best to intervene right then and there, but if you can't, at least half an hour before or 1 hour before, not any time later it becomes the gray zone of whether it has happened or not happened. So, clearly synthesis of new proteins, synthesis of proteins de novo is very important here, and you are stopping, when you stop that, the memory does not seem to be getting consolidated. This is yet another proof that how a behavior like memory retention can be traced down to the levels of a molecule, right. Proteins are molecules, and we have not zeroed it on one particular protein here, or but we have done a blanket blocking of the new proteins, but the point is you need to be able to synthesize new proteins to be able to store this memory for a long time. When I say this, and then when I give these lectures, people typically ask that, so can we trace down all this behavior, all this memories to molecules, and if it is the root of all this behavior, it is hard for us to realize the fact that things as complex as such behaviors can be traced down to individual molecules. Are there not any other explanations, or are we missing something here? Clearly the answer is that yes, you can trace down to individual molecules, no doubt about it, but these behaviors have multiple scales, they are operating at multiple scales. While my point here is to stress on the fact that you do need these molecules to be able to express this behavior, they by themselves are not just sufficient, and these kind of so called the plasticity occurs at multiple scales. The scales can range from molecular scales that, as we have seen, at the individual proteins, or even down to dopamine levels or neurotransmitter levels, or it could be at the level of synapses, where the efficacy of the throughput, like synapses are the places where the neuron A communicates with neuron B in the brain, and then the efficacy of this communication could be a way of storing the plasticity. And at the pan neuronal level, that the response of a neuron for a given stimuli could be altered, right. The general excitability of the neuron through expression of different ion channels and so on and so forth. Apart from this, it could also be a network level phenomena. However, what we will see is that we, from the vast literatures we had in this field so far, we could say much of this could be traced back to one of these three, not to say that the network do not play a role, but then even those network level phenomena could in some way be directed to one of these three levels, neuronal, synaptic or molecular levels, and that is the line that we are going to do in this lecture at least. Well, if they are operating at multiple levels, then what evidence do we have to say that it is at the synaptic or even at the neuronal levels? So, while behaviorists and psychologists were very busy figuring out what are all the rules that govern the learning in human beings and mammals and in animals in general, neuroscientists, trying to understand the functions of the brain and how it relates to the behavior, were busy trying to establish the physical correlates or the physical relationship between the function and the organ itself, the brain itself. In one of such experiments, Bliss and Lumo, they were trying to understand the role of neuronal circuits at the hippocampus itself. Remember, hippocampus, the removal of the hippocampus is in HM resulted in loss of declarative memory. Now, using that as a cue, they really wanted to probe on to this effect of onto this hippocampal region, and then see if they can actually zero in on some correlates of plasticity. So, now so here when you are looking at that kind of a region, I mean that kind at that scale, then we are specifically looking at changes in the input output characteristics of a region or input output characteristics of a neuronal circuitry that you are looking at, or even a pair of neurons that you are looking at. So, in order to understand their experiment and then what their question really is, we need to understand a little bit about the hippocampus region itself, specifically the neuroanatomy of the hippocampus itself. So, now let us take a look at the mice brain, and the mice brain when we look at it, and that is the rostrocaudal axis, the nose or the head is in this direction and the tail is in this direction of the mice. You can think of that the mice being here, and then you are placing the nostrils of the mice were to be here, and then the tail were to be here, and you extracted the brain. That is how it looks like. Now, you can look at this brain from the top when you look at it, that is how it looks, right. So, same again the regions, the directions are the same. So, that is the head or the nose, and this is where the tail of the mice is situated. In this and this axis is called as the rostrocaudal axis. Now, when you are looking at the anatomy of the brain, we are going to look at the multiple sections and then say, and then look at how the neuronal circuits are arranged here, neurons are arranged and how, what are the kind of connections that they are making. Especially we are going to look at hippocampus, all right. So, when we look at the hippocampus, when you are talking about sections, then this any 3-dimensional structure for that matter, particularly this 3-dimensional structure, can be taken sections at various different ways, and 2 routine ways in which people take sections are along 2 mutually perpendicular axis. So, when you place the mice brain, orient the mice brain in this way, and if you take a plane that is perpendicular to the rostrocaudal axis, see that is this is our rostrocaudal axis, right, head to tail axis, let us call it as head to tail axis. So, if you take a section that is perpendicular to the head to tail axis and then flip it around, so that and then clearly the other axis would be parallel to the head to tail axis. So, let us say if you take a section perpendicular to the blue line and then take a section and flip it around, so that we can actually see it in a plane, and that is how it looks, this coronal section. I am pretty sure if you have been watching these lectures, this section is very familiar to us. When we looked at Hitchum, the picture next to him was a mice brain, and I wanted to illustrate why Carl Lashley failed in his attempts to localize memory, right, and to reject your memory. I even said that is the region called hippocampus, and that is the region that equivalently in HM that has been removed, it is mice. So, HM in HM's case it was human brain, and medial temporal lobe was removed, which included the hippocampus. On the other hand, if you were to take a section along the red line, along the head to tail axis, and then look at it, and that is how it will look like. Okay. There are various different regions which are marked here, and the sections that you obtained on an axis perpendicular, on a plane perpendicular to the head to tail axis, we call it as coronal section, and while the other section which is perpendicular to the coronal section, we call it as sagittal sections, and the region that we are particularly interested is in this region, hippocampus. So, for various different reasons, it is easy to look at the circuitry of the hippocampus in these sagittal sections. So, when you look at it, the sagittal section of a hippocampus, what you are seeing are subdivisions, sub-regions of the hippocampus. Now, just going back here. So, whether you look at it in this direction or take a section this way, that would be the sagittal section, because of the 2 overlapping C sections, right, 2 C's, you can think of that as this going up and down, and then if you take a section here and this way, they look very similar, all right. So, and that is how it looks in the sagittal sections, and this hippocampus then is divided into 4 main regions in this section, region of which 3 of the regions are called as CA regions, coronoaminous regions, and they are called CA1 marked here, CA2 not marked here, and CA3 again marked here, and dentate gyrus is also called a CA4 or the dentate gyrus regions. Each of these regions have a marked difference in their cell types, and they have a very characteristic input output relationships. So, the input to this hippocampal region comes from cortex entorhinal cortex through a pathway called perforant path. Okay. So, these are the perforant path, and the axonal fibers that coming in, they are called as the perforant path fibers or perforant fibers, and then they come and then make synapse into the dentate gyrus cells, and the dentate gyrus cells make synapses, send their inputs to the CA3 neurons, and the output of the CA3 then goes into CA1 pyramidal cells. So, since it is a very fixed neuronal circuitry involving 3 synapses, synapse number 1, 2 and 3. It is also called as a trisynaptic circuit, and it is quite extensively studied. It is very uniform, and they have very remarkable characteristics. Number 1, because it is very uniform, you can actually structurally say where exactly the fiber bundles are, like they are very distinct, very easy to visualize, and then thereby you can actually, if you want to stimulate, you can electrically put in an electrode in a region and be sure about that you are actually stimulating the fibers and not the inputs and not anything else. And you can also be, because the cell body layer and so on are separate, you can also be sure about placing an electrode in the right place near the cell body, and be sure enough that you are recording at the output end of the neuron. Thus, having the pretty regular anatomy helps to be able to dissect out the circuit behavior, and it helped Bliss and Lomo to perform the experiment they much needed wanted. What is that experiment? They said, hey look, if the hippocampus is important to store memories, and we think, I mean memories we think are something that we form in response to events that are happening around us, there has to be some plastic change, some change that has happened because of an event and that stays just the way it is, and the event, thereby the event has an ability to leave its mark in the brain even after the event has happened. So, that ability to retain to leave a mark in the brain, we call it as plasticity. It is plastic, like a plastic, you can mold it and then that stays there, unlike an elastic where you release the force and it collapses back into its original state. Here it is plastic, so it stays there, so that you can go back and then retrieve that memory. If that were to happen in hippocampus, and particularly if it were to happen in through the synapses, then why do not we make use of this architecture, internal architecture of the hippocampus, and then architecture, I mean the neuroanatomy of the hippocampus, and then probe, does it really happen. So, how do you probe it? What they said is I am going to implant 2 electrodes, one a stimulating electrode and a recording electrode. So, the idea is that we are going to stimulate the neuron and then record the electrical activity, okay, and see what happens to the electrical activity in response to the stimulation. Does the brain, the hippocampal region, has an ability to store this fact that there is a stimulation that has happened in time, and if it stores, what is the expression of this storage? How do you see that, in what form does it store? Now, these were the questions that they are asked by Bliss and Lomo, and we will in the next lecture see the exact experiment, how they did it and what do they see. Thank you.